So welcome back in the second video lecture of construction and screening of genomic DNA library and cDNA library. Okay, so first, what comes in your mind when I will ask you about a library? Not just forget about the genomic DNA library for some time. Just think about library. What comes in your mind? So you will think about a large room that will contain a couple of books, right, of various subjects, of various authors, right? Similarly, genomic library means it contains a collection of genes, okay, and this collection of genes will be organ specific, meaning the meaning is each of the genomic library is constructed on the basis of a particular organism. That means you can heard about E. coli genomic DNA library, okay. You can also heard about human genomic DNA library. That is the idea, okay. That means on the basis of a particular organism a genomic DNA library is constructed, okay. So now what is present in this genomic library? So as a normal library consists of every kind of books, right? Similarly, a genomic library of a particular organism will obviously contain the all of the genes that is present, or all of the DNA fragments that is present in its genome, right? That means with the help of or by constructing genomic DNA library, the idea is to preserve the genes of a particular organism, okay. That is the idea of genomic DNA library. That means we are collecting the all the DNA that is present in a particular organism, okay. So now let's think about this. So what to do? What will be the procedure by which we can make a collection of genes? So there will be a procedure known as in vivo DNA cloning, right? Because in vivo DNA cloning, if we are not lysing our host, then the DNA will be remain within the host, right? That is the idea, that on the basis of the normal DNA cloning that we have learned earlier, on the basis of which the genomic library is constructed, okay. So one thing is clear, we have to clone the entire genome, right, so that we can take a record or take a collection of genes, right? So now the question is, can we clone the entire genome of a suppose E. coli or any kind of higher eukaryotes with a one vector? That is quite impossible, right? So we need multiple vector for that. So our first step for constructing genomic DNA library will be the cleavage of the entire genomic DNA with a mixture of restriction enzyme, okay. So that will give us a multiple fragments of genomic DNA, okay. Although here I have described about only 1 2 3 4 5 6, but there can be many, okay. So now the question is, why I have used a mixture of the restriction enzyme? So the answer about this is to generate overlapping fragments. So now what will be the utility of generating overlapping fragment? So now let's see this one. Suppose we have applied restriction digestion, we have cloned each of the fragments with the help of a particular host that I am going to explain you after that one. Then we have plated it in an appropriate media. Okay, so now why we clone, why we will construct a genomic DNA library? The idea is very simple, so that we will construct genomic DNA library so that we can study about their genes later on when we require, right? Let's take example of the recent SARS-CoV-2 virus, right? So there is already a viral genome library for SARS-CoV-1, right? So during the first few weeks of the pandemic, they have compared the genomes that they have preserved for SARS-CoV-1, and that on the basis of that they find out that this SARS-CoV-2 is quite different from the SARS-CoV-1, right? That is the idea. That means whenever we need them, we can study them. That is the idea why we are preserving or take a collection, or we are taking a collection of a particular organism's entire genome. Okay. Similarly, let's say about a human genome. Let's say there is a particular genetic disease that is observed in a particular individual. Okay, so if we record the entire genes collection from that particular individual, and then in future if those genetic disease reappeared in another individual, so we can compare and find out that yes, this is because of that particular gene, for example X, right? So that is the idea. Whenever there is sudden pandemic, so whenever there is various kind of mutations, right? For example, in coronavirus, in case of coronavirus or COVID virus, we have seen about the Delta variant, Omicron variant, right? So that variants organisms can be stored in a library, right? So that we can use them for further study, right? That is the utility of genomic DNA library. It is helping you to study an organism's entire genome whenever you need. Okay. So now, as you want to study, so let's say about a particular genome, let's consider about the example of E. coli, then it will be easy to understand for you. Okay. So suppose you are willing to analyze, or you willing to study for about the lactate gene. Okay, that is the important gene coding beta galactosidase of lycopene, right? So now let's think about this, that among that particular colony, although in reality there will be more colonies, what colonies contains cells, that cell contains your particular lactate gene, right? So it is quite difficult to understand, right? So under such circumstances, the genome sequence that is for lactate gene will help you to find out your gene of interest. Okay, so one thing I want to clear, that if you don't know about what is the sequence of a particular genome, then it will be very difficult for you to screen a particular gene of interest from a particular organism genomic library. Okay, and now suppose you want to sequence an organism's entire genome. Suppose there is a organism X whose genome is completely stored in the laboratory, in the library, sorry for that, laboratory. So now you have to screen them. For screen them, the first thing that you have to do is you have to sequence them, right? So for sequencing, as you can see that in that procedure we are cloning each of the fragments, and then we are transferring them a particular host, right? And then we are plating the host cell containing cultures, and then we are getting colonies, right? So can you clone them? Can you sequence them but just by looking the colonies? No, that is not possible. So under such circumstances, you have to lyse each of the colonies cells, okay, so that you can get nucleotides. Then you have to attach them to get the complete DNA sequence, right? That is this one. So now let's consider two situations. For example, you have applied a mixture of recombinant DNAs in a mixture of restriction endonucleases, and you have obtained two fragments. For example, just taking an example, A and B. Okay, so overlapping means the A fragment will contain some sequence which will be common with the some sequence of B. That you can see here, the nucleotide stretch of nucleotide GCATTA is the common sequence that is present in both A and B. Okay, so that will help us to understand that yes, the sequence B is just after the A. That is why we have some overlapping part, right? So that is the utility of generating overlapping fragment. That means whenever you are requiring the entire genome, you can easily get that. But if you use a specific type of restriction endonuclease, then we will end up with something like that I have mentioned here, that is the fragment number C and fragment number D. Is there any kind of overlapping region? No. So now you tell me that can you provide any kind of logic with the help of which we can say that the sequence D is just after sequence C? Is it possible? No. That is why whenever we are requiring the entire genome from the genomic library, so if we cleave them, if we cleave our entire genome with the help of a mixture of restriction endonuclease, then in future this cleavage with mixture of restriction endonuclease will help us to construct the entire genomic DNA again. So that is the idea. Okay, so I hope now this is clear to you. Now what will be, so we have generated a number of multiple overlapping fragments. Okay, so we will now insert them into a cloning vector. Okay, now we will introduce them into host cell and facilitate cloning, and we will end up with this thing. Okay, now let's say suppose that those cells are present in a conical, and then you have to plate them in an appropriate LB agar media, and you will end up with colonies. Okay, these colonies, each of the colonies in the plate will contain clone of cells that is derived from a single ancestor cell, because during incubation time the cell is dividing, cell has divided its number, cell has increased its number, right? So each of the colonies will contain a millions of cells which are nothing but ancestor of a single cell. Okay, and each of the cell will contain a particular stretch of nucleotide sequence. That means in each of the colony we have multiple copies of a particular nucleotides, right? So that is how we will end up with a number of recombinant colonies that will together construct or contain the entire DNA fragments that is present in the genome of E. coli. Let's say here we are constructing a E. coli DNA library. Okay, that means a set of recombinant colonies which contains the entire DNA sequence of a genome will known as genomic DNA library, or will construct the genomic DNA library. Okay, so idea is very simple. Our goal is to simply preserve or collect the DNA of an entire genome. Okay, so as we cannot load the entire genome of the organism in a one vector, we have to digest them with the help of a mixture of restriction in the nucleus that will help us by generating a mixture of different overlapping fragment, right? Then we will inserting our gene of interest in a cloning vector, and then we will clone them in an appropriate host, like, okay, then we will plate them in a LB agar medium, okay, containing appropriate antibiotic. So each of the colonies will contain clone of cells derived from a single ancestor cells, and therefore it will harbor a single copy of a particular nucleotide sequence or particular stage of the fragmented genome. Okay, so that is the idea of the genomic DNA library. In that way, you will end up with a set of recombinant colonies that will together contains the entire DNA fragment of a particular genome. Okay, so that is the idea of genomic DNA library. So in the next video, we are going to discuss about how during the screening procedure, because I have told you the main goal of constructing genomic DNA library is so that we can study about a particular gene in future when we are requiring, right? So suppose you have to study about LAGZ gene. So before studying about LAGZ gene, the first goal should be you have to find out about which colonies will contain the clone of cells that contains LAGZ gene, right? So during this screening procedure, how many recombinant colonies you have to screen? Okay, so I think you may say that suppose the entire genome of recombinant entire genome of E. coli is 20,000, okay, and then our average insert size of the vector is, suppose, let's say 20, okay. That means there will be 1,000 of different cells or clones we have to screen to find out our gene of interest, right? But this is not the actual number, because the actual number will always be greater than this 1,000, because there can be a chance of overexpression and not expression here. There can be a chance of that some of the DNA fragment will be over represented and some of them will be down represented. Okay, so for be safe, we have to construct in such particular example that I have given to you more than 1,000 colonies to find out the gene of interest. Okay, but there is no kind of formula until that part, that how many clones we have to analyze to find out our gene of interest. Okay, so there is a mathematical formula that helps us to find out how many clones we have to discuss or we have to screen to find out our gene of interest. We will going to see in our next video. Okay, and I want to mention another procedure, that is during the study of an entire genome you have to screen them. So the name of the screening procedure is colony hybridization or plaque hybridization. Okay, so we will also going to see about colony hybridization and plaque hybridization in our upcoming videos. Okay, so that was the concept of genomic library, why we have to screen genomic library, and how we can construct them with the help of in vivo DNA cloning. Okay, and why we are using a mixture of the recombinant DNA, mixture of the restriction endonuclease, so that we can generate overlapping DNA fragments. Okay, and what is the utility of the overlapping DNA fragment in the genomic DNA library. Okay, so that was the video for genomic library. So I hope this video will be helpful to you, and if you like this video, please hit the like button, share with your friends, and subscribe to my channel to get more videos like that. So we will again come back with another video lecture of this chapter, where we will going to see the formula that will help us to find out how many recombinant colonies we have to screen to find out our gene of interest. So till then, thank you for listening to this class, and bye bye.